Hi everyone, welcome to Physiology for Students. In this lecture, we're going to talk about inheritance, the process of passing down traits from parent to child. In this lecture, we will review the human life cycle and the production of gametes through the process of meiosis. We will also talk about some terminology related to inheritance. And then we will discuss some simple patterns of inheritance related to genetic diseases. Inheritance is the passage of traits from parent to child, or the parent generation to the offspring. This was classically studied by Mendel, and so sometimes you'll hear the term Mendelian inheritance for simple patterns of inheritance of genetic traits. Mendel classically studied inheritance of parent generation to offspring. He studied pea plants, other animals, and he's considered one of the fathers of human genetics. So first, let's look at the human life cycle. I know a lot of this is probably familiar, but we're going to use some of this to apply to the genetic terminology, so let's just make sure we're all on the same page. So, for the parent generation of human, we have the female, which has ovaries, giving rise to the female gamete, which is the oocyte, or the egg, and the male of the species, which has the testes, or a singular testis, which gives rise to the male gamete, which is spermatocyte or sperm. When the sperm fertilizes the egg, then both combinations, maternal and paternal chromosomes, combined to make a zygote, which has chromosomes from mother and chromosomes from father. That zygote then grows into an embryo, a baby, and the baby then grows and develops into the adult human, restarting the life cycle all over again. So you'll notice I use the word gametes. Gametes are reproductive cells that are produced through the process of meiosis. It's important to note here that for the human life cycle, gametes each have a single copy of each type of chromosome. That's called haploid, or half the number of chromosomes that are in the adult of the animal. So that is 1N, or 23 chromosomes in each haploid gamete. So again, you have the maternal chromosomes in the oocyte and the paternal chromosomes in the sperm. When they come together, you have one copy of chromosomes from mom, one copy of chromosomes from dad. Once they come together, then you have a diploid, or two copies of each type of chromosome, a total of 46 chromosomes in that zygote. Then, of course, those cells divide, continue to develop to produce the embryo, baby, etc. So let's review a little bit of meiosis. We're not going to get into every single stage of meiosis. So most of you have already had meiosis in your prerequisite courses for your now clinical courses. If you need a review of meiosis, I'm going to point to, in our diagrams, the OpenStax textbook, which has a really nice set of diagrams and descriptions if you need more detail. So meiosis is the process of gamete production, ultimately forming these haploid gametes in the female, eggs, and in the male, sperm. So what's important about meiosis is that you have two complete rounds of nuclear and cell division. So last lecture we talked about the cell cycle and mitosis. Mitosis is the process of nuclear division followed by cytokinesis, cell division. In meiosis, you have two complete rounds of that, so that you end up splitting the groups of chromosomes in half, and that's how you end up with haploid gametes, with half the amount of chromosomes in the original cell. Another important piece of meiosis is that the chromosomes actually touch and exchange genetic material. This is very important for allowing the mixing of genes. As those chromosomes are split apart, they have the opportunity to mix up genes so that you'll have a mixture in each set. And that's why I can have my grandmother's nose, my grandfather's ears, my great-grandfather's lips, etc., because things get all mixed up, very different than what happens in mitosis, where things are very faithfully copied and sorted. Here, they're intentionally mixing genes across the chromosomes. When the chromosomes touch, that's called chiasma, or crossing over. There's also assortment of chromosomes, so that maternal and paternal homologs of the chromosomes in that parent cell are going to be separated randomly rather than exactly faithfully on each side of the cell the way that they are in mitosis. Again, that's why I can have my grandfather's one thing, my grandmother's other thing, because they're all mixed up. Okay? So it's very important to understand that not only does meiosis allow for haploid gametes, so you only have a single group of chromosomes, but also that those haploid gametes have genetic variation from the original parent cell. They're different. Now, if you've gone through other biology courses, you've probably talked about the importance of genetic variation from parent generation to offspring generation and the implications of these small changes in genetics across each generation. Thank goodness we're not exactly like our parents, right? No, I love you, Mom and Dad. So let's take a second to compare meiosis and mitosis. So I'm going to zoom in on this diagram here so you can see the differences. So hopefully what you can see here is that there are 2 rounds of cell division in meiosis 1 and then in meiosis 2, compared to mitosis, which is just a single round of cell division all the way through to the end, cytokinesis. You'll also notice that there are places where the chromosomes touch and mix up genes. And here they're drawing that in these sort of blue and red different combinations of genes, where you're seeing now you have some blue and red on each chromosome, as opposed to always blue or always red. They're mixing up the genes. That does not happen in mitosis, because in mitosis you want exact copies of the parent cell. And then another thing I want you to notice is that at the very, very end of meiosis 2, you end up with a single copy of each chromosome. So those are haploid cells rather than the original diploid cells. Okay? All of this splitting and mixing of chromosomes in meiosis can lead to the potential for chromosome abnormalities in the gametes, which get passed on ultimately to the baby. So chromosome abnormalities are common, and they can lead to, unfortunately, common causes of mental disability in the offspring and miscarriage or a non-viable pregnancy, depending, of course, on the chromosome alteration and the genes that are involved. One of the most common types of chromosome abnormalities is a whole chromosome non-disjunction, meaning that it didn't fully split. So instead of having one chromosome on each side at the end of meiosis, maybe the gamete ended up with 2 chromosomes. Let's say that was in the egg. If the egg had 2 chromosomes and then combines with the sperm that has 1 chromosome, you end up with 3 copies of that chromosome. That's called a trisomy, or 3 chromosomes instead of the usual diploid. Now, you could also imagine how you could end up with no chromosomes, no copies of a certain type of chromosome in the egg cell. And then you would have a monosomy, meaning that you'd only get the chromosome from the paternal line. It's not usually all of the chromosomes, it's usually just one of the 23 chromosomes. That would be a monosomy, or a single copy of a particular chromosome in all. There's also chromosome breakage, translocation, and certain fragile sites on chromosomes, all leading up to various disorders that are related to chromosome abnormalities. Now that we've covered a little bit about meiosis and the formation of gametes, I want to talk you through some of the terminology so we can then use it to understand various genetic conditions. So, first, locus. Locus is a term that we use to talk about the location of a particular gene on a chromosome. If we're studying a particular gene that's involved in a genetic disease or a particular trait, we can actually trace it down to where it is exactly located on which chromosome, and that's called the locus. Remember that genetic inheritance is determined by an individual's set of paired chromosomes, and then we're going to look on each chromosome for that gene at a particular site or locus. For example, we talked about red hair. I know I love to talk about red hair, so this is an easy one. So the gene for red hair is the MCR1 gene located at chromosome 16. Then we get specific as to which exact type of gene is carried on that chromosome. So red hair is an easy example. There's many different types of hair color. So the allele is the specific form of the gene that is present on each chromosome. So on the maternal chromosome, you can have an allele for red hair, black hair, brown hair, etc. On the paternal chromosome, you also have the possibility for red hair, black hair, brown hair, etc. Okay. So which type of allele is on each chromosome tells us then what are the possible traits that could be inherited in the offspring. So let's get a little more specific. As I said, MCR1 is a key gene for pigmentation. There's two major forms of the MCR1 gene. There's eumelanin and pheomelanin, and we write those as a single letter. We write a capital R for a dominant trait, or the trait that will dominate if it's present, and that for hair color is brown or dark hair. So we write a capital R for pheomelanin. And then red hair is what we call a recessive trait. It's going to be hidden in the background if the dominant trait is there. So if pheomelanin is present, it covers up the gene for red hair. So that's a recessive trait. We would signify that with a small r or a lowercase r. So R lowercase will be for eumelanin, which if present would give red hair, but it will only show red hair if there's no pheomelanin present, okay? So, if you have the same type of allele from the maternal side as the paternal side, we call that homozygous, or the same type of allele. So that could either be a homozygous RR, capital RR, or a homozygous lowercase rr, meaning that both parents carry the same type of allele. If each parent is different, then you have heterozygous parents, meaning that there's one copy of the dominant and one copy of the recessive, and that would be an R, big R, little r. I'm using R here as an example, but in genetics, we can use any letter for the gene that we want to. You might use A or B or whatever. Now we get to two very important terms, genotype and phenotype. Genotype is the genes that are carried within an individual. Phenotype is whether those genes are actually expressed or what it looks like. So the genotype, in my case for red hair, would be little r, little r. I had to get little r from my mom, little r from my dad. Now I have both copies of that recessive trait. So I have the genotype little r, or eumelanin. The phenotype then is the description of what it actually looks like. And in my case, that is red hair. So when I'm at the grocery store and I'm with my kids and all of us little redheads are running around, and there's always that really nice old grandma that will say, "Oh, those cute redheads, where did they get it?" And it depends on the day for me. There's some days where I'm like, well, from their mom, of course. And then there's other days where I'm a little bit crabby, or maybe I'm just feeling super scientific and I'll say, well, it's a recessive allele, so I got it from both of my parents and then I passed it on to them, but of course they had to get it both from me and their dad, so we're just a whole redhead family. I know, I'm a nerd. Okay. A little bit more about expression of a gene. It's not always this simple, so there actually can range in levels of expression, and we call that the penetrance across the generation. So the penetrance is the percent of individuals with a certain genotype that express a certain trait. So how strong is the expression of that trait? And then there's also age-dependent penetrance, and that is individuals with a certain genotype may not actually express the trait until later in life, and we certainly see that with some genetic diseases and other conditions. In science and in many other areas, we use genograms to follow the inheritance across multiple generations of families. Some people call this a pedigree, and then we can formalize it into different types of genograms. So there are medical genograms where we're actually tracking certain diseases like diabetes and hypertension and breast cancer down through families. And this is where genetic counselors can come in and help to look across generations and actually now sequence the DNA and see what traits you have in your genome. So a genogram, though, is a simple graphical representation of a family tree across generations. This is a silly joke. Of course, what I'm showing you here is the genogram of Anakin Skywalker, which I'm sorry guys, I cannot get the Star Wars families straight. Every time I go see these movies, my husband has to explain it to me. But those of you Star Wars fans, here you go. Here is the genogram of the Anakin Skywalker family tree. But we could make this kind of a family tree for traits or genetic diseases as well. When we look at genograms, then certain patterns of inheritance are evident, and we can then actually tell from those patterns where the likely disease is located. So we can actually tell whether it's on an autosomal chromosome, 1 through 22, or a sex-linked chromosome, X or Y. We can also tell whether it's recessive or dominant. So let's look first at autosomal. So autosomal diseases are caused by mutations in genes that are found on the autosomal chromosomes, numbers 1 through 22. If they are autosomal dominant, that means that one copy of that gene is sufficient to cause the trait or cause the disease. That gene dominates over any other particular alleles that may be present, kind of like we talked about for brown hair and red hair. If the gene for brown hair is present, it covers up the red hair, and you'll never see the red hair. You will only see the brown hair. So dominant means that trait will be expressed over other allele possibilities. So if present, the dominant allele is going to be expressed. Autosomal recessive is hidden, meaning that you have to have 2 copies of a recessive trait for that trait to actually show up. And this is good news for certain genetic diseases because 1 copy of the allele is not sufficient. However, it does mean that if you have only 1 copy of that disease that you can carry, you're a carrier, you can pass it on to your children. So often when we look at autosomal recessive traits or diseases, we say they skip a generation, meaning that the carrier generation doesn't show the trait. But if 2 carriers get together and each pass on their recessive alleles to their offspring, then the child can have that trait. And that's the case in my family. Actually, both of my parents have dark hair. So they were carriers for the red hair, meaning that even though they had dark hair, they carried the red hair gene. And then each of them were able to pass on to me their recessive copies of the red hair gene. And then I became the redhead. So recessive genes are hidden by the dominant genes. They're there, you just don't see them. And then those individuals are carriers, meaning that they have the allele and can pass it on to their children. So that means you need to have 2 copies. If it's a disease-causing gene, you need to have 2 copies of the recessive allele in order to actually express that disease. So let me give you an example. For autosomal dominant, if it was big R, little r, big R, big R, either of those genotypes could lead to the dominant phenotype. But in autosomal recessive, a big R, little r would be a carrier and would not have the disease. Only the little r, or homozygous recessive would actually express the disease. We have a simple way of predicting the probability of any particular trait being passed on from parent to offspring if it has a simple inheritance pattern, meaning that it fits these Mendelian style autosomal dominant and autosomal recessive, or sex-linked dominant, sex-linked recessive type inheritance. So for a Punnett square, you're going to draw a box with 4 squares, and then you're going to draw the parent genotypes from parent 1 and parent 2 and cross them. Let's say, for example, that both parents are carriers for red hair, and we want to know, is that baby going to be a cute little redhead? So we would say if they're carriers, that means they have 1 dominant and 1 recessive, big R, little r. They would have the phenotype brown hair, carriers for red hair. Same with parent 2, we'll say that he is a carrier, he has brown hair, big R, but he carries the red hair gene, little r. So what's the probability that any child will have homozygous recessive, or that red hair genotype with the red hair phenotype? Now we simply cross the boxes. So these first 2 go in the first box, that's big R, and then these go in the next box, big R, little r, and so on. This box now is going to be big R, little r, and then the last possible combination, little r, little r. So you can see in this example with heterozygous carriers for a recessive trait, 1 in 4 probability for each child that they would have homozygous recessive or little r, little r. What is the probability that they will have the dominant trait? That is anywhere you see big R. So there are 3 out of 4 cases here that have the dominant allele. So 3 out of 4 have the possibility for brown hair. So we say 3/4 dominant, 3/4 have the dominant gene, which can lead to the dominant phenotype, and 1/4 are homozygous recessive, which will lead to the recessive phenotype. That's the way to do the Punnett square. Try a couple of different combinations on your own. Try a parent combination where one parent has both recessive alleles or one parent has both dominant alleles and see what sort of probabilities you get in the offspring generation. Now in this diagram, I'm showing you some other Punnett squares for autosomal inheritance patterns. So take a look and pause here to check out the possibilities for autosomal dominant and the possibilities for autosomal recessive. And the terminology that we use is affected parent, affected offspring, unaffected parent, unaffected offspring, meaning that they don't have the disease, or carriers. And remember that carrier is only used in terms of recessive because they carry the gene, but they don't express the trait. Now let's look at sex-linked diseases. Sex-linked diseases are caused by alterations in genes on the X or Y chromosome. Notice that the Y chromosome has very few genes. That means that if there is a disease on the X chromosome, the Y chromosome in some ways acts as a recessive allele, meaning that it does not protect against a disease on the X chromosome. So males and females will be affected differently. So remember, in females, you have 2 copies of the X chromosome. Let's say that the mother carries a genetic disease on the X chromosome. So the mother has the genetic disease, but the father's chromosome can then protect against it. So if that genetic disease is a sex-linked recessive on the X chromosome, the mother's allele has the disease, but the father's X chromosome in females can protect against the disease on the mother's X chromosome. If, however, the mother X chromosome has a recessive trait, genetic trait, but it's a male, the Y chromosome doesn't have the gene to balance out that recessive trait. So, same mother, more males of the offspring generation will be affected than the protected females. Okay? So, females have 2 copies of X, and that second X can protect them against a recessive trait on the X chromosome. There's also X inactivation, which means that only one X in each somatic cell is active and the other becomes a bar body. That means you have a random distribution of X types throughout the body. So not all of them will be expressing the diseased allele. In males, however, you have one copy of X and they are unprotected by the Y chromosome. That means that the males of a particular generation, if the mother carries a disease on the X chromosome, it's much more likely for males to get the disease than for females. So the big clue that you have an X-linked disorder is that you have a lot more males than females affected by the disease. Here are some examples of the sex-linked inheritance patterns. I'm going to show you the first one, and I'll let you take a look at the others. The first one is the most common example, and that is where you have a carrier mother who doesn't have the disease but simply carries the disease, and an unaffected father who has a normal X chromosome coupled with a Y chromosome. In that case, look at the percentage of males versus females that are affected by the disease. The females are going to be here with the XX. They get one X from mother and one X from father. Half of the females will carry dominant alleles from mother and father. Half of the females will get the affected gene from mother and be carriers, but none of the females will be affected by disease. Now let's look at the males. Half of the males will get the dominant allele from mother. Remember, mother was just a carrier. And then the Y chromosome from father, which doesn't change their phenotype. And then half of the males will get the affected chromosome from their mother and be affected by the disease. So notice the difference between expression of the disease in females versus males. Half of the females are carriers, but none of the females are affected. Half of the males are affected, but none of the males are carriers. The males are either affected or not in this case. Okay, 2 more examples. I'll let you pause and go through those other 2 examples, see if you can figure them out. Okay, this all underscores the importance of family history when you're talking to your patients. And this is why in every encounter you talk about family history when you ask your patients their history questions. You want to know, are there certain conditions that run in their family? Then you can start sort of building that genogram in your head of what you need to be looking out for that particular patient, given their family history. So the question then often comes from the parents that says, will my children have this disease? This disease runs in my family. Are my children going to have this disease? And that is where the importance of family history comes in, and the importance of the inheritance patterns of those diseases. So if you hear from a patient that they have a certain disease in their family that you know, for example, is an autosomal dominant disease, then yes, there's a high probability that their children can have that disease. If, on the other hand, it's a recessive trait, then you can dig a little bit more deeply to see, yeah, it's a lower probability for that particular disease. So you can begin to then counsel patients. And of course, if it's more complex, that's when you would then refer them to genetic counseling. All right, I hope that was helpful. You can refer to OpenStax biology, meiosis, and inheritance mainly for this lecture, although there is a brief bit of this in Guyton chapter 3. Feel free to add any questions in the comments below. I love getting your questions and I'm happy to answer them. And if you'd like to stick around, we'll have some challenge questions coming up.